Well, good morning. Welcome to a very special colloquium. I've known Chris Johnson for a long time. You're all in for a treat. I always enjoy his talks, and I always learn something from them. Chris has a very distinguished bio and history. And I promise to only do the short part. Otherwise, we'd be here for a long time. But he is the Distinguished Professor of Computer Science and the founding director of the Scientific Computing and Imaging Institute, cleverly the SCI Institute at the University of Utah. He also holds faculty appointments in the departments of physics and bioengineering. His research interests, of course, are in scientific computing and scientific visualization. And it's one of the wonderful things about the SCI Institute is that it really is an institute of scientific computing. We often see these visualizations and associate the visualizations with Chris, but it is a scientific computing institute. He's a fellow of the AIMBE, AAAS, SIAM, and IEEE. And he's been awarded the IEEE Visualization Career Award, the IPDPS Charles Babbage Award, and the IEEE Sidney Fernbach Award. And with that, I will turn it over to Chris.
Thank you, Bill, and thanks everybody for coming today on a nice Friday morning. It's beautiful weather out there. Congratulations to the Cubs. And I almost had that as my first slide. I thought, that's how I'm going to get the most applause. So before I get going with the talk, I always like to give a little bit of history about visual computing in Utah because most people don't know about it. This guy on the left is David Evans, who was brought in by the president in the mid-'60s to start a computer science program at Utah. He came from Berkeley, and the first person he hired was Ivan Sutherland, which was a great choice. Ivan had created the first computer graphics program called Sketchpad. He would later win a Turing Award for his work in graphics. The third faculty member was Tom Stockham, and Tom was a signal processing guy who invented the CD-ROM, amongst other things. He also created algorithms that helped unerase the Watergate tapes, and which is very interesting. And then those three faculty with a few others really created this environment by which they had amazing students and that created a lot of computer graphics that we use every day. So some of our graduates are John Warnock. John co-created PostScript and then co-founded Adobe and then was the longtime CEO of Adobe until just recently. Number 5 there is Ed Catmull. Ed got his PhD at Utah. He had then co-founded Pixar. He's the president, has been the president of Pixar, still is, and now the president of Disney Animation as well. Number 6 is Alan Kay, and Alan co-created the laptop computer and also object-oriented languages. He won the Turing Award a few years ago. 8 is Jim Clark. You probably know about Jim here, and Jim got his PhD at Utah and then went to Stanford for a little while and then founded Silicon Graphics Incorporated, SGI, and then Netscape, and then Healthion, and then WebMD, and then my CFO, and he's currently the CEO of Commandscape. He'll be visiting me in Utah in a couple weeks. We're looking forward to the visit. This is Nolan Bushnell. Nolan got his bachelor's degree at Utah. He created Pong, the first video game, and then founded Atari. And he tells the story of when Steve Jobs and Steve Wozniak worked for him at Atari, and that when Steve Jobs came up to him and said, "Hey, Nolan, I want you to invest in this company I've got called Apple." And Nolan said, "I don't think so." I didn't think he'd make it, and Nolan then founded Chuck E. Cheese Pizza Time Theaters, which have been popular as well. So, and then lots of other people, if you're a graphics person, Jim Blinn and Garreau and Fong and others that have come through there. So it's really amazing. And so we have this legacy of visual computing at Utah. And these are the current faculty at Utah. They are mainly in these 3 areas, which are information and scientific visualization, biological, medical, and geophysical image analysis, and scientific and biomedical computing. And we pretty much have 5 or 6 regular faculty in each of these areas, plus research faculty, research scientists, postdocs, and then 80 to 90 PhD students spread across those 3 areas. Just to give you a sense of our interdisciplinary nature, these are the national centers that we direct ourselves, a long-running NIH Center for Integrated Biomedical Computing. Valerio Pascucci directs a DOE Center for Extreme Data Management Analysis and Visualization. We're one of the NVIDIA GPU Centers of Excellence. We have a Center for Computational Sciences that Russ Whitaker directs, a new DoD Center for Computationally Designed Efficient Materials. And we're part of a number of other national research efforts, these 3, for example, in DOE. Okay, so let's go. This is your brain. It's a simplified version of your brain, thanks to Tor Nortranders, that divides it up based upon your different senses, approximately in terms of size and in speed. So as you see, sight, our visual systems, take up about half of the volume of our brain. It's also the fastest part of our brain, followed by touch, and then hearing and smell, and then taste. We are very, very visual creatures. And the idea for many of us in the visualization world, all of us in the visualization world, is that if we can find the right mappings, the right abstractions from this large amounts of data, complex data that we're creating, that this seems to be a reasonable way to better understand that data. So we spend a lot of time trying to figure out what those new mappings are, efficient mappings, appropriate mappings, and abstractions to take very complicated data and understand it. From galaxy simulations to the compaction of new materials. This is one of the largest combustion simulations done in the world by Jacqueline Chan. Some stellar magnetism here. This bottom one is very interesting. It's work done with Valerio Pascucci, one of my faculty, and researchers at Berkeley and Lawrence Berkeley National Laboratory, where they were trying to compare the latest experiments in fluid dynamics with the latest theories and computations. And they were looking for ways to accurately and quantitatively compare those different experiment and theory and computation, going from laminar flow to turbulence. And Valerio figured out over 5 years of working with these scientists a way using topological analysis where he could actually count bubbles during phase transitions and then relate that to experimental and theoretical computational numbers. So it was a big paper in the journal Science, and it was a really great example of this long-term collaboration of understanding the science and creating new visualization methods to solve an important problem. We have confocal microscopy. We have some jet fuel set on fire and doing the fundamental chemistry and physics on very large DOE supercomputers. We have some both experimental and computational fusion going on. And more often we're seeing data that is very high dimensional, but not necessarily a 3-dimensional spatial relationship. So for example, epidemiology, spread of disease, comparative genomics, molecular biology, and others. Okay, so let's do some case studies. We've done a lot of research in the field of volume rendering, and volume rendering is a visualization technique to take 3-dimensional scalar data and then figure out how to bring interesting features and parts of the volume up to the screen. And the traditional methods of volume rendering are that we have basically you take these rays that go through the screen in parallel, and then you add up the contributions of the image intensity or some scalar value along that ray, and then you relate the image intensity to a color and an opacity or transparency. So this is called the transfer function, and it's the color and the alpha, and then F would be the image intensity. Well, these guys, my colleague Chuck Hansen and former students Gordon Kindleman, who's now at the University of Chicago as a professor, and Joe Niss, who's at Google, figured out a way to generalize this to higher dimensions. And so instead of just looking at only relationships to the image intensity, their first version was they could also look at things like gradients, which would look at the surface changes in materials, or curvature, They've now, Chuck and other students have now extended this to higher dimensions to look into parameter spaces too. And what this allowed us to do was to look at scalar data in new ways and see a lot more specificity. And so this is just a CT scan of a tooth. And it's the same data, but then using a three-dimensional transfer function, we are able to then pull out a lot different views within that same data set just by changing that transfer function. So this is my favorite example of why it's important to have these higher dimensional transfer functions and also to be able to do very large scale data. So this is the NIH visible male. He's the most famous dead guy in medical imaging. And after his death, his body was donated to the National Institutes of Health, and he was scanned with all the highest resolution medical scanners of the time, MRI and CT scans, and then they froze the body, placed a color camera on top, took a photograph, and then milled down the body every millimeter, photograph, et cetera. They put all of that data online and made it publicly available, so he became the first gold standard of human anatomy. And the data from his body has been used in literally thousands of research projects and image analysis publications. The data sets were so big, and this was done 10 or 15 years ago now, the data sets were so big that no one could visualize all of them. So what they did was they downsampled the data, and you got images that look like this. Because we had the ability to visualize the entire data sets interactively, and we had these high dimensional transfer functions, we found out he was covered with tattoos. So this is not a photograph, this is a volume visualization of the ink in the skin where the input data is a set of photographs. So if you think about it, the input, we've got a photograph, set another photograph, and if you think about what a small scale feature the ink from a tattoo is in the skin, and then the ability to pull that out, you might not be surprised to find out that people who'd worked on this data set for 10 years or more had no idea he was covered with tattoos, because the first thing they did with the data was they downsampled it. And it was such a small scale feature, it went away. That really got us thinking about, given this day and age of big data, that the knee-jerk reaction is always, well, I've got so much data, I will just downsample it and do dimensional reduction before I even consider looking at it or analyzing it. Well, be careful, because if you have small scale features that might be important, you might be just getting rid of all those. We could even see a little red in the flag. It says mom across his chest here. So it was pretty interesting. We now have this system that runs on mobile devices like iPhones and iPads. That's a 256 cube CT scan of a hand being fully rendered on the GPU and CPU of the iPhone. And for me, it's amazing, because it was not that many years ago that was a $30,000 graphics workstation to do that, and now it's in our pockets. And this is really opening the doors to a tremendous amount of new possibility for large-scale visualization in mobile environments. And one of the applications we've been working on is in medicine, and multiple applications. A specific example I'll show you is with my colleague Chris Butson, who's a computational bioengineer and also in the departments of neurology and neurosurgery. And he's trying to understand the fundamental effects of and causes of movement disorders like Parkinson's, and also their resulting treatment with deep brain stimulation. So patients that have tremor that they can't control with drugs, there is a treatment now called deep brain stimulation. And this is a surgery where they're implanting these electrodes, and they're trying to find the place in the brain they can stimulate to disrupt that current that's causing the reentrant current. And if they get it right, they go from not being able to drink or feed themselves to near normal movement again. It's actually miraculous to watch this patient who was not able to hold steady that cup, and now will be able to go back to a near normal life. So where do we come in? Well, we come in after they go home and they heal. Things move around a bit, and they have to come back to the hospital to have the stimulation electrodes optimized. And the current way they do that is they hook them up to a system and they watch the tremor. And then they just start with electrode 1 and stimulation site 1, voltage 1, and they watch voltage 2, and they watch, and they go through everything. It takes 4 to 5 hours for them to do the optimization. What Chris does is that he creates an image-based model, a computer model, from the patient's scans, MRIs and CT scans of the head and the brain and where they have inserted the electrode. And then he does large-scale finite element simulations of all the possibilities of every electrode stimulation site and their values. Once he's done, we've made a version of our volume visualizing system that runs on an iPad, and he sends a text message to the neurologist, who can then come in with the patient and connect this up to the stimulators. And then within less than 5 minutes, he's able to they are able to optimize where the right place is. So it's using this guided visualization that's from simulations, image-based modeling simulations of the patient-specific anatomy that enables them to do this so quickly. So he just got a large NIH grant to do a clinical trial at the major DBS centers in the country at Stanford and Cornell and the University of Florida. So it's really exciting. I think this is a really great example of where high-performance computing and high-performance visualization, and then taken down to the mobile devices, is going to change the way we do medicine. All right, this is another example of
FlowRender is a tool that was developed for confocal microscopy data, and it's focused on giving biologists an easy-to-use method for doing visualization and analysis of 3D confocal microscopy data. So Gabrielle Cardin here at the University of Utah has found that FlowRender is a valuable tool for her research in the Cardin lab.
The Cardin lab works on congenital defects in muscle development, such as congenital diaphragmatic hernias. We're using it to image developing limbs and developing diaphragms, which are very complex three-dimensional structures. So I would say that we would be unable to do any of our work unless we had FlowRender.
That's what we like to hear from our collaborators. I was talking with Donna Cox a lot yesterday about how visualization is changing and really making new science available, and so I wanted to put in a few of those examples for you today. This is another example we're working on with Nasir Marouche, who's a cardiac surgeon specializing in atrial fibrillation ablation, and this is the case when the atria, the two small chambers, are fibrillating. And in the past, so it's not generally thought of to be life-threatening, in the past they would primarily give drugs for this. But now they're finding that there's a significant increase of stroke possibility if you have atrial fibrillation. So the current best treatment is to be able to take a catheter up near the heart and then find the places that are causing that atrial fibrillation and ablate them, which means taking a electrical discharging and actually killing cells that might be causing that circuit to cause the fibrillation. And we do a similar thing than what Chris Butson does on the brain. We do this with the heart, starting with an MRI, and then creating patient-specific models to do simulations and then visualizations of the places where Nasir, we think, would be good places for him to ablate. And as you'll see, this is a real surgery. He spends his time just staring at these 7 30-inch monitors the whole time. To do these surgeries. He does multiple of them a day. He's gotten so good at it, people are flying in from all over the world to have their surgeries done by him. And because it's been so successful, a company has just bought the technology to take it through the FDA. So it's a really great example of visualization and scientific computing really impacting people's lives on a daily basis. All right, this is another one from the science side, where high dimensional visualization and coming up with the right abstraction was key to better understanding of new science. So this is from a combustion simulation, and the idea with a lot of these combustion simulations and studies is trying to find more efficient fuel mixtures. And they have all these different chemical species that make up these dimensions from the simulations, and how do they best understand what combination of those chemical species will give the most efficient combustion. And because it's so high dimension, 10 dimensions, I know of no one that can reason in a 10-dimensional space. Some of us can reason in 2 and 3 dimensions pretty well. Even that's challenging. But once you start going 4, 5, and above, it really becomes impossible to accurately understand those high-dimensional spatial interactions. So what do most scientists do? Well, they break out those 10 dimensions in a set of 10 2D plots. So this is the 2D plot of hydrogen over time, and O2, and then oxygen, and OH. And then they look at all 10 of these, and they watch the different curves, and then they try to infer the interactions from those sets of 2-dimensional plots over time. And even that is really, really hard. Well, a couple of my researchers, Valerio Pascucci and Ross Whitaker, along with graduate student Sam Gerber, came up with a new 3-dimensional abstraction that was basically a way to combine all of these 10 dimensions into something 3 dimensions. And we'll show you what it looks like. Let's see, there we go. So we're going to put this into place here, and you'll see there's a little bar down here. That's the time bar. And then when it moves this, you'll see all 10 dimensions move over here. And the researchers knew about 2 of these arms for the different mixtures of the chemical species, but they didn't know about the 3rd one. And so what this new abstraction gave them was really new insight and understanding to some of the fundamental science underlying combustion. And so this is an example of, if you can find the right abstraction, the right mapping to this really complicated high-dimensional data, that it can provide insight, new insight for scientists and engineers. All right. Let's do some more high resolution, high dimensional data. So this is a really wonderful data set that Mark Lavoie from Stanford University a few years ago convinced the Accademia Museum in Florence to give him full access to Michelangelo's David statue. And he used a high-resolution laser range-finding scanner to scan the statue at sub-millimeter resolution. He then gave out the data to the visualization community to say who could visualize this, and we could. This is using a parallel ray tracing system, and we're showing you the level so you can see the individual triangles there. And we could also interactively change the lighting and shading, so when the statue is outside and the model is outside, you could see what it looked like at different times of the day. So that was pretty cool. But what is cooler is that he recently resampled and scanned the statue at much, much higher resolution. So now each of these images you're seeing is 2 gigapixels in resolution, not megapixels like your phone, but gigapixels. And it's almost 1 billion little triangles. We're going to zoom into that same place in the eye, and at this resolution, you'll see sculptor's marks and defects in the marble, but you can't see individual triangles. It's that good of a resolution. It's amazing. It is such a high resolution and so good now that the Accademia Museum will not let Mark give out the data anymore. And we are not allowed to show except 4 different views, because if you had a big enough, high resolution enough 3D printer, you can now print out your own version of Michelangelo's David statue, which is exactly what they want in case something ever happened to the statue, and also to enable further study of the statue. The technology behind being able to do this interactively is Valerio Pascucci on the right. He has a system called Vis3D, and there are special data structures that use space-filling curves that were published a couple of years ago at the supercomputing conference that enable him to do this large-scale data streaming. This is something we didn't know about, was that somebody has carved their initials, M and N, on the back of one of Michelangelo's David's legs. we're pretty sure you can't get that close to the statue to do that now, but at some point, probably when it was outside, someone did that. The ability to render.
What would be wrong with 3D printing it? I mean, why wouldn't they want that?
I think that it's their intellectual property, and they want to decide who controls that intellectual property. So they might sell the rights to somebody, a limited number of museums to do that, but they just don't want everybody to be able to do it. Because if you go there and look at their gift store, there's a lot of little Michelangelo models that they want to sell you.
I recommended to some museums that they do exactly that, provide 3D modeling so people could 3D print
Yeah.
the great, antiquities and so on.
Yeah.
Oh.
Well, talk with them and see if they'll let you do it. Anyway, and so parallel ray traced system to be able to actually render this. And we have now recently released a iPhone and iPad app called Gigapixel David that you can find on the iTunes store. And you can see the 4 different views that we're allowed to show you interactively at that resolution on your phone or iPad. It's really quite amazing because they're really gigapixel images interactively done on your phone and iPad so that you can see the same different views. So it's free. Download it today and play with it. I left this in here, although yesterday I was treated to these amazing visualizations from Donna and Bob on astronomical and astrophysical data. And but I went ahead just to put this in here because it's from a really large-scale simulation that Elena Dongnia did when she was at Harvard. And the story is nice because she was at our Department of Physics and Astronomy, and she had just done the simulation of the formation of the spiral galaxy, which was the largest ever done, at least at that time. And she had no way to visualize her simulation data. It was many terabytes of data. And our physics people told her about us, and she came up and met with me, and I said, I think we could visualize this. And so the next week I got a FedEx of all of her data, her disks, and many terabytes, which took a long time to read in. And then we used this parallel ray tracing system we've created called Manta that was able to treat each one of those little stars as a sphere and do the correct lighting, shading, and coloring based upon the physical parameters of her simulation. And it really created some just beautiful visualizations, and we have other ones that I think are also very compelling. It was certainly at the time, it may still be the most accurate simulation. Then she was able to then compare it with the observation models. But it's still not as beautiful as what Donna and Bob have created. So go, if you haven't seen their stuff, you should go see it. It's amazing. Okay, back to some big image analysis and visualization. This is part of the Human Connectome project, and it's an international collaboration amongst neuroscientists who are using mosaic electron microscopy to try to understand the fundamental wiring diagrams of our brain. So what they do is they've got these amazing technology to slice the smallest piece of the brain, image it at the highest resolution of electron microscope, and then dump it out the disk. Slice, image, dump, and they do this 24 hours a day, 7 days a week. And this is one image, mosaic, to look at. And so if you see this little scallop here, for example, that's the size of the maximum electron microscope. So that's 4,096 pixels by 4,096 pixels. Remember, your humongous HD TV is only 2,000 by 1,000 pixels. So it fits right nicely in that little side there. So when they do this, and it doesn't come out like this. So we have image analysis registration tool. Tolga Tazizan, one of my faculty, has created a set of open source tools that do this. When it's done, one image is 200,000 pixels by 200,000 pixels at a resolution of 2 nanometers per pixel, 2 times 10 to the minus 9th meters per pixel. That's small. And then they do the next slice and the next slice. And they cannot slice in the depth, of course, as nearly to the resolution as they slice across in the mosaic way. And so even though they're slicing as thinly as possible, you see that there's quite a bit of change. And so this makes the tracking problem very challenging. And we've already gone through at least 2, maybe 3 dissertations on that. And I think there are more to come. So once they get done with this, they get a volume of 16.5 terabytes for one little volume. And then they give it to us and they say, okay, we'd like to interactively visualize that now. And we can do it. It's a combination of the Vis3D data streaming systems and then our image Vis3D parallel volume visualization system. So that's a power wall that has 36 30-inch monitors and 41 megapixels of resolution, and we can interactively visualize that 16.5 terabytes on that wall. What's really interesting was that the neuroscientists spent hours and hours looking at this data, and because no one had seen this data at this resolution, they made new discoveries by looking. So it really, to me, was like a new microscope. Robert Mark, who's the neuroscientist who created the techniques for slicing, and then Valeria Pascucci's Vis3D software, the streaming, and then our ability to volume rendering this, it gave them a new window into this data that they had never been able to see before. And it harkened back to those days of when the first microscopes were created, and they discovered a lot of new science because they could see things at resolutions that no one else could see. And so in a sense, I think the combination of high performance computing and visualization along with these new techniques in neuroscience and data streaming enabled us to create a new microscope for these neuroscientists. And this is what one of those remember this is 2 nanometers per pixel, and this is only one cell type in that little volume. And it just, when I see this, it just strikes me as how interconnected and complicated our wiring diagrams are within our brains. And I think that the scientists who are doing this, and they're each taking different parts of the brain and doing the same type of technology, and then the idea is they're going to connect them all together and create a full wiring diagram of the brain. It's going to be very challenging. That's at 2 nanometers per pixel at that level. Just last Friday, a week ago today, Brian Jones, who works with Robert Mark in the lab, and I and some others put together, just submitted a proposal for the next level of technology for this. So they now have the way of doing 100 terabyte per volume data sets at higher resolution, and they have a path forward to 300 terabytes per volume. And thinking that these are going 24 hours a day and creating these things, it already stretched us to do 16.5 terabytes interactively. We don't know how to do 300 terabytes interactively. It's also the case that not only do they want to visualize, but they want to be able to simulate. So they want now to try different bioelectric models and biochemistry models and other searching algorithms. And because of the data set size, those all become high performance computing problems. So it's a tremendous challenge for the future. Okay, so let's get to some information visualization. This is Mariah Meyer. She's one of my young stars who does information visualization. Unlike a lot of the visualization researchers that we work with, who go after technique-driven research and will often find new techniques to do vector field or tensor field or whatever kind of visualization, and then just kind of you get your data and munge it into their format and try to use their system. She actually really works closely with the scientists. In this case, she worked directly with scientists at the Broad Institute, which is a Harvard and MIT collaboration, and she embedded herself into the laboratory. She read the papers of the scientists, interviewed the scientists to find out their goals, used their tools, found out what worked and what didn't work in their current visualization systems, and then she went away and designed a system that she thought would solve that particular problem better. And she put her system in the hands of the biologists and let them use it, and then got feedback of what worked and didn't. And then she did another version, and then same. And so this rapid iterative prototyping in the way of designing a better visualization system. She has now done this many times, and here are 4 of her visualization systems. And you notice that they're different looking, and that's because they solve 4 different biological problems. And I'll give you one fun example. This is with some comparative genomicists at the Broad Institute who had a model comparing the chromosomes of human that are represented in this outer ring with the chromosomes of different mammalian species, which are represented in this inner ring. And this is the first time they looked at their model data in Mariah's system. The broad red bands and green bands here are correct. All of the rest of this is incorrect. So they knew they had a problem with their model. They went away and worked on their model, and this was another version of it. It was better, but after seeing this, they knew they had something fundamentally wrong with their model. They then went away for months and using Mariah's system until they came up with this version of the model, which was experimentally verified and then published. And Mariah asked the PI of the lab how long he thought it would have taken him to get to this point without her visualization tool. And he said, quite frankly, I don't think I ever would have gotten there. And it's just such a great example of a visualization researcher who's working closely with a domain scientist and being able to solve a problem that neither of them could have solved before by effective collaboration and interdisciplinary collaboration. And I wanted to give you a sense of how important interaction is. So you've seen some stills of the system, but it really is an important system, important to have this interaction. So they have many different types of data, and they're asking many different types of hypothesis and testing them and wanting to visualize the results of their hypothesis. And so interaction is key. So they spent hours and hours and hours in front of Mariah's visualization tool until they were finding clues that led to them to a better model. And you might not be surprised to find out that Mariah now has a line outside of her door of people who want to work with her. Okay, so this is some work that's done by Alex Lex, who's another of our young information visualization faculty, and he's been working on a number of things, including a lot of work on cancer and visualization systems and pathways and other things. I thought I would show you at least a couple of other information visualization systems that he's worked on. This one is called Upset, which is about set visualization. And most of us, when we think of sets, we go back to the day when we were first introduced to sets, and we did everything with Venn diagrams. You remember this? You have a circle and another circle, and then we had the union and the intersection and other set operations. Now think about those set Venn diagrams when we have more and more sets. Instead of 3, think about, say, 50. If you have 50 circles and you want to understand their relationships, how well do you think you could do that with Venn diagrams? Not so well. Now think you've got 5,000 or 100,000 or 5 million. So the Venn diagram method of visualization does not scale very well. And it is actually a very challenging problem to figure out how to do set visualization. And Alex has created the system by which has a multi-window integrated approach to doing set visualization. And I have a little video that does have sound. So just to give you a short view of the set visualization. Let's see, maybe I have to.
Upset also visualizes elements and attributes. To demonstrate this, we have loaded a dataset about movies that contains movie genres, release dates, ratings, and popularity. The view here is aggregated by sets. Here we can see box plots of the attributes: release date, average ratings, and times watched. In the element view, we see a scatter plot of release date versus ratings for the whole dataset. It seems like ratings in the past were higher, but this is possibly a bias of the data collection. We can see that the number of movies in the sets is quite different. To get a better understanding of the size of the smaller bars, we can adjust the scale. Bars that break the scale wrap around themselves to keep them comparable as long as possible. Let's take a look at the documentaries. The box plots reveal that documentaries are rated higher on average than other genres. Documentaries also don't seem to mix much with other genres. Selecting their element reveals that most documentaries are recent productions. The war genre, which is also highly rated, contains a lot more classics. Looking at the intersections, we see that war movies are more often of multiple genres than documentaries. We hope you enjoyed our tour of Upset.
So just a quick view. Another of Alex's work that won the best paper award at the Information Visualization Conference a couple years ago is on rankings. We all see these US and news rankings of various departments and universities and everything, which to many of us academicians really is frustrating. What Alex and his team did was to create a system by which you could decide what was important to you amongst the schools. So it could be the faculty citations, it could be the job opportunities, it could be many different things, and you could put the weight in that category and then be able to see the rankings based upon your own setup, and you could compare these to others. And just as a quick view of what this looks like, of how the rankings, the system works, maybe not. Let's see, I'll let it. All right, it's not cooperating, that's all right. We'll move on, sorry. Okay, some of my own research is on uncertainty visualization, and I always start this with the motivation of when is the last time you've seen an error bar in a three-dimensional visualization, including mine? And the answer is almost never, maybe never. And I use the motivation of the example of tumor segmentation for pre-surgical planning as an example of why I think it's important that we include this kind of uncertainty information and error information. So this is an MRI. It's a little dark here, but let's see if I... the way that we do this pre-surgical planning is that we go around the edge of the tumor and we segment, and then each of the slices, and then we build up a isosurface that represents the tumor. And you'll see there's no indication of uncertainty here. Well, if we look at the sides, on this particular side over here, it actually looks like an algorithm where a person could approximate that edge. But there are other places, well, it's really dark, other places on the image where you really can't tell. And if you take different segmentation algorithms and apply it to that data set, these are the extents of those different segmentation algorithms. And so on the left, you could say, we have, say, high confidence of the approximation of our surface here for that particular part of the tumor segmentation. Over here, however, we have less confidence, and maybe this is kind of approximates a confidence interval of the size of the distance away from each other that give them a sense of the uncertainty when they're doing their planning for surgery. So it's my contention that we should be doing this kind of uncertainty visualization for all of our scientific visualizations and information visualizations and images that we're showing people to help them make better decisions under uncertainty, because all of the decisions we are making are under uncertainty. And just because we don't show it doesn't mean it's not there. It really is. So let's try to come up with accurate and useful mechanisms by which to look and visualize that uncertainty. So one example of a system we worked on to do this is for diffusion tensor imaging. And diffusion tensor imaging is a type of magnetic resonance imaging where we can use multiple gradients to move the magnets in the MRI around in a way that we can recover the underlying structure or direction of the structure of the white matter tracts. So this is a dissected part of the brain where you see the corpus callosum and then you see the white matter tracts taking sensory information out to the cortex. One reason this is important to know is in surgical planning, for example, tumor resection, where the tumors can often move the location, normal placement of the white matter tracts away, or sometimes even envelop the white matter tracts. This is a beautiful visualization by Ron Kikinis at Brigham and Women's Hospital and annotation. This took months to do. And unfortunately for surgeons, our brains are not color-coded and mapped like this. So when you open up people's brains, they kind of look like gray jello. And so the surgeons do not want to cut through these white matter tracts because depending on where those are, it could deplete some part of our sensory information and apparatus. So you'll notice there are no indications of error on that. Well, there are errors. And this is now a little bit old, but it will give you a sense of, if we look at the actual data resolution that we have for, that's a raw diffusion tensor image. And as we come through, we'll put a little box glyph on there to show you the size of the voxel that we're using as input. And you'll see they're pretty large. And what we're doing is taking those voxels and then we're interpolating, and we make these beautiful images over here of these white matter tracts. Well, neurologists don't believe these. They publish them in their papers, but they don't believe them when they're planning to, and neurosurgeons don't believe them either. So we put together and did an analysis on different algorithms for recovering these white matter tracts. These are two different algorithms with the same exact data set, and you can just glance at them to see that they're different. And so one of my students worked to create a way to be able to visualize both a comparison of the algorithms, and we also worked with our carbon fiber folks to create a phantom for diffusion tensor imaging so that we could have a ground truth. And then we could look at these different algorithms compared to the ground truth. Blue is low error, red is high error. And then we have across this interactive glyph here, we have a histogram that shows an estimate of that error as you move through the data. So it was a first step to be able to try and better understand and convey this type of imaging error and uncertainty. I want to give you a quick glance before I finish on just a couple of very new things. So this was just published last month, 2 months ago. It's work with Hanwei Shen, who's one of my former PhD students, who's now at Ohio State University. And it gets to the problem of three-dimensional dense vector field visualization. So in vector field visualization for 3D, we often have these lines, these streamlines that represent the vector fields. But as soon as we have a large scale system in 3D, it just looks like a bunch of spaghetti that you can't understand. So we tried to use interaction and deformation as a way to help better understand this problem. And we now have a way where if we have a mathematical description of the features, for example, a vortex, we can say, find all vortices in this field, and it will just open up a window surrounding that vortex. We also have ways that you can interactively move through the data and layer by layer go in and zoom in to be able to look around in the data. And you still see the context overall of where you are in the data, but now you have a window into some of the features that you might find that are interesting. In addition to using this for vector field visualization, you might not find it surprising that this could be useful for looking at diffusion tensor images in the brain or other places in the brain. And so we have now started to create a system by which we could show neurosurgeons for pre-surgical planning again how we are able to go from outside to inside, layer by layer across those white matter tracts. So here's an initial example of opening up those white matter tracts layer by layer to better understand and reveal the underlying tumor that's there. We're working on a better interface and more control, and have gotten feedback from the surgeons. Okay. And going back to uncertainty, and then that clip and slide I showed you about Chris Butson's work on the deep brain stimulation, we just published a paper on the uncertainty visualization of the different methods from different surgeons using different locations to apply that stimulation protocol for the patients. When Chris started to interview a number of these neurosurgeons who were doing deep brain stimulation, they all thought they had the exact right place and placement of the electrodes. And so he captured those and with stereographic methods, and then he put them all together in the same view, and it looked like a bunch of pickup sticks. So they were just every which way. They were close, they're around the same area, but they really weren't exactly the same by any means. And so we're creating a visualization system for him so that he can better understand the variation of those different placements of the stimulator electrodes and convey that information back to the neurosurgeons and see if we can now better understand which of those are more successful than others. Okay, so I'm going to end here and show you the smiling faces of the 200 faculty, staff, and students of the Scientific Computing and Imaging Institute. And these are our productivity machines. We have 5 espresso machines in case 4 break down. I saw that right across the hall here, you have some nice espresso machines being introduced, and I absolutely know that the next time I come back, the productivity is going to be soaring here at NCSA. We also have a ping pong table, foosball table, exercise equipment, etc. One of the funny things I learned when we put out this ping pong table is that ping pong is apparently the international nerd sport because everybody from all over the world plays ping pong. So the people from China, India, Brazil, Germany, Italy, America, etc., they all seem to play ping pong. So we have a tournament every year, and then I play the winner. So far, I've only won 1 game out of those games. Anyway, all right. Well, thank you for your attention. I will like to see all of you in Salt Lake City in less than 2 weeks. Thanks very much.
We have time for a few questions for Chris.
Yes, Gabrielle.
I was wondering if the graduate students that you have working with you, do they come in with the skills that they need to work on these problems, or what is the kind of training and courses that they need to take?
That's a great question. So I would say in general the answer is sometimes and usually not. The types of work that we do often requires a particular level of mathematics, and the computer science students in particular, since I was an assistant professor, it's just been take away this math course, take away this math course, take away this math course, to where they have a very, I think, poor or limited mathematics background. So we've actually had to create courses on the math you should have learned as an undergraduate that you will need to do graphics and visualization and image analysis and scientific computing. Also, we have 2 different PhD tracks within the School of Computing at Utah. There's the traditional PhD in computer science, which is theory, architecture, systems, etc. And then we have a PhD in computing, and that's track-based. So we have a PhD in graphics and visualization, and one in scientific computing, and one in robotics, and one in imaging, where the students really come in and focus on those areas in the beginning, and they don't need to have a PhD in computer science. They can have PhDs in math or physics or other areas, and they might have to take courses, but they often come in with, like, the physicists and mathematicians have much better math skills, for example, but they might not have some of the computational skills that they have to learn. So it's this mixed bag. It's very rare that we have students who come in and just have a great background because it's not the usual typical things they learn as undergraduates. So they often haven't had courses in scientific computing or image analysis or visualization, and so there's often needed learning that has to be done along the way. Yep. Yes.
So immersive VR is coming to the consumer space. How do you see that impacting what you're doing over the next few years?
We did quite a bit of work in early days of some of the stereo and immersive. We created something called the haptic workbench several years ago, and then back in those days the resolution and especially the tracking just wasn't fine enough to be able to accurate enough to do the kinds of work that we did, and so we kind of went away from that. But now I think we are seeing, I was talking to Donna yesterday, we were seeing this tipping point that I find very similar to when the graphics days of, the $30,000 graphics workstation and that each triangle costs several dollars, down to when the 13-year-old boys wanted to play shoot 'em up games, and with the commoditization of the graphics into these cards in which really triangles and pixels became free. And I'm seeing the same thing now in the augmented and virtual reality arena, where it used to be very expensive, custom designed systems that are now coming down to the commodity and the consumer. I don't think they're quite ready in terms of accuracy and resolution, but I think that the more 13-year-old boys playing games with them will drive that kind of development, and that we will see in the next few years, this tipping point where that's going to be able to be used for science, engineering, medicine, and daily activities, games, et cetera, on a much broader usage. So I do see that we're about to that tipping point. It's going to be really interesting to see how that develops.
Any other questions? Let's thank Chris again.
Thank you.